It is time for us to get started this evening. Welcome to Life Science Cafe. My name is Elise McCormick, and I'm a first-year PhD student in the Organismal and Evolutionary Biology program at UMass, and I will be your MC this evening. So a little bit about Life Science Cafe. We were established in 2011. Life Science Cafe is organized entirely by graduate student researchers at UMass. And every month we invite experts to share and discuss their research. Typically, we'd like to host these events here in the Amherst community, and we're so excited to see so many people here with us tonight. This is amazing. But we're also continuing to have a hybrid format. So we also have this available for people on Zoom. And we'd like to take a moment to thank our funders, the IDGP program at UMass, the Graduate Student Senate, and the Graduate School at UMass. Today, we are very excited to have Dr. Jess Gersony with us here to talk about her work on tree ecophysiology and the junction of science and art. Dr. Gersony got her bachelor's degree at Columbia University, completed her PhD at Harvard University, and conducted a NEPA-funded research program as a postdoc at the University of New Hampshire on a project titled Understanding Drought Sensitivity in Eastern U.S. Forests to Inform Sustainable Forest Management Practices. So we are so excited to have you here to be able to talk about trees.
Thank you. I'm very excited to be here.
So just a quick primer for you and for our audience as well. We're going to have 3 vignettes tonight with a pause for audience questions after each vignette. So we're going to start with vignette 1, getting into some basics about trees. So first, can you break down for us what makes up a tree and why is it different from other plants?
Yes. Hi, everyone. So I primarily study trees. I've done a few projects studying plants that weren't trees, but trees are where my heart is. Well, the biggest thing that's very different is they're huge compared to other plants. And so they have a lot more stresses on their system because they are so much bigger. And so they need to be photosynthesizing a lot more. They need a lot more water than smaller plants. And they need a lot more area for roots and nutrients in the soil. So that's, I think, the biggest thing is their size. And so inside of a tree, you can see over here. Oh, I think maybe there's a laser. Maybe not. Oh yeah, maybe. Yeah, perfect. Okay. So in a tree, they have these piping systems, and that's what I studied. So I studied the xylem and I studied the phloem. And the xylem is the blue pipe, which is for water. And the red pipe is the phloem, which moves carbon, which is what I did my dissertation on. And what the reason why we were studying trees is because there's a bunch of research on smaller plants, because you can grow them in greenhouses, you can grow them in growth chambers, you can grow them outside, you can just grab a leaf off of them. They're much easier to study. But the trees are so important in thinking about the regional carbon cycle, which is the primary reason why I was excited to try and study them and see how their insides are different than smaller plants and how they deal with stress differently than smaller plants. Does that answer?
Yeah, that totally answers my question. So could you give a little bit more of a breakdown? And you just talked about phloem, but why is phloem so important?
Team phloem for life. Okay, so phloem is very important. So it's pretty much, we call it, some people call it, I call it the sugar highway of the plant. And so what happens is photosynthesis is happening in the leaves and it makes sugars, and those sugars need to go all over the plant. They need to go to the roots, they need to go to the growing shoots and growing fruits, and even in the stem, they need to go everywhere so the plant can respire, because plants respire like humans do. They break down sugar to make energy. And so sugar is needed everywhere all the time, but it's primarily being made in the leaves. So the phloem is what allows for that sugar to move all around. And what's really cool, I mean, I can go on forever, but I'll try to say like one word, 2 things. So the phloem, it's really important because it's this main transport system. And so if it lost functioning due to drought or due to some other stress by climate change, it could potentially cause the tree to die. Because if you're photosynthesizing, you can photosynthesize all you want, but if you can't move the sugar to where it's needed, the plant will die of hunger. And so it's really important. But despite it being really important, we really don't know anything about phloem functioning in tall trees. We don't even know how much sugar is in it. And I should say, a lot of people are surprised by that fact because they're like, what about maple syrup, especially in this area? But maple syrup actually comes from the xylem, which is this crazy thing that the xylem does in the spring. Yeah, I know. It's amazing. My students were, yeah, it was very fun. We tapped trees this season, which was really fun. But yeah, so we don't know a lot about the phloem, and we don't know a lot about it because it's really, really small. So these tubes are like 10 times smaller than the xylem tubes. They're on the order of like 10 microns. And I think there's something about like a human hair is like 100 microns or something. So they're like 1/10 of that. So they're very small. So that's one thing. And so it's hard to say just objectively because of that, but also because the trees have these like healing processes like humans do. So if you puncture the phloem, or even if you touch the phloem, if you touch the plant, it can be so sensitive that it stops functioning. It like clogs it up in the same way we are like white blood cells clog up our blood veins. It's like similar to that. And so if you cut a phloem, typically all these proteins will clog it up and you won't be able to get any sap out of it. So it's like this defense, which is great for the tree because main people trying to invade the phloem are bugs who eat the sugar. And so it's a way to protect them against bugs, but it's also protecting them against researchers. And so the researchers have been struggling. So it's been like a big methodological issue for a century, essentially trying to figure out what's going on in the phloem.
That's amazing.
I love it. That's why I've dedicated a lot of time to it.
That's so cool. So kind of trees as a whole, what would you say is their kind of special importance or role in ecosystems? And what is something that maybe people here would be surprised to find out about trees' roles?
Yeah, okay, great. So I love these questions. It's like all my favorite things. Okay, so I think I would say two things. So I think a lot about gas exchange. So I think a lot about carbon going into the plant for photosynthesis and water coming out of the plant because the stomata are open. And I should give a nod to Angie Patterson, who is in the house. And Angie taught me, she was my plant physiology TA at Columbia. She did her PhD at Columbia. So most of what I'm saying actually comes from Angie's brain. But yeah, so I mainly study gas fluxes. And so two things that I think are really cool is one, their role in the regional carbon cycle. So in the northeastern US, we emit a bunch of carbon like everywhere through our anthropogenic CO2 emissions. And the northeastern forest, depending on what study you're looking at, can uptake between 20 and 40% of those anthropogenic emissions. That's huge. Like that's a huge, like when I first read that, I was like, oh my God, this is no joke. These forests, they're really doing work. So they're really important for the regional and also the global carbon cycle. And then, so that's one thing. And then the other thing, they're also really important for the water cycle, which is, I think, something that people think typically less about because it's not as directly related to climate change. But one of my favorite facts is, okay, I usually say it's a question, what do you think? But I'll just tell you. So, and like a big mature red oak tree, so there's probably one around here somewhere. They're very common. They transpire or they have water moving through them every day, and the amount of water that moves through them in a single day is 100 gallons of water. One red oak tree. So they are pumping water out of the ground and putting it back into the atmosphere. And so that's another really important role of them. Those are my two big carbon and water.
That's amazing. Yeah. Oh my gosh, that's so cool. That's so cool. So obviously you love trees.
Yeah.
But your kind of focus within science is ecophysiology. Could you explain for us what ecophysiology is and how you study it in trees?
Yeah, it's a great question. Okay, I think I have, let's see, I don't quite know what the next slide is. Great, okay, cool. Okay, so ecophysiology is the study of physiology or like the functioning of a plant. So it's like, how does photosynthesis work? How does water move through the plant? Same thing as like anatomy and physiology of a human. It's like that, but for a tree. So just thinking about like, how do things move inside of you and how do you create energy? And then ecophysiology is thinking about how the ecology and the environment around the plant influence that physiology. So it's really kind of at the intersection between ecology and physiology. And the reason why I'm so passionate about it is because I got into this work thinking about climate change, and I wanted to really think about how trees are responding to the changing environmental situation around them and how we might be able to use them or like lean on them to take care of us through this crisis. But also try and understand like what's going on for them and like trying to communicate that to the world, like, no, they're struggling, this species struggling more than that species because of this mechanism and that mechanism. So trying to get at that kind of level of detail into how, like, is the maple dealing with this super differently compared to the oak, compared to the pine. And yeah, specifically thinking about a lot of these gas fluxes, so thinking about CO2 uptake and water loss, and how you study it. I don't have a picture of Angie, but so the way that you study, you have to get to the top of the trees usually. This is really fun. But it's pretty hard to do. So for my dissertation, I got trained on using this bucket lift at Harvard Forest, which I think goes about, I think it's like 100 feet or 130 feet. It's very, very tall and very, very fun. And so you drive it, you get up to the trees and you sample the leaves. That's one main way to like get your sample to analyze it. The other way is shotgun sampling, where you bring a shotgun into the forest and you shoot down the leaves and you catch them as they fall, like rain, and then you put them in a bag and you bring it back to the lab and they analyze them. So it's a lot of field work. So you usually, to get at the ecophysiological questions of the forest, you got to go to the forest, which is part of the reason I love it so much. And so you get the samples and then you can analyze them in the field. So this is me doing a pressure bond measurement, which is a measurement of how stressed a tree is for water. But then you can also bring it back to a lab and do further like chemical analyses on it, or microscopy, or any different types of tests on it in the lab.
That is so cool. I had no idea that you could do any of those trees. That's amazing. So this is obviously out kind of in the forest, what people would normally think of. They're going to think about going to study trees, but you also have some research that focuses on urban trees. Have plants kind of adapted their phloem for urban living? And if so, what about that have they altered?
It's a phenomenal question. You should come work in my lab. So we don't know the answer to that. So I just started the urban work when I moved to Smith. So I got here, I started in July. So not even a year ago. Yeah, brand new. Loving it, by the way. Great place. But so yeah, I started when so I guess the story is when I was doing my PhD, I was doing this type of work. And when I was doing my postdoc, I was doing this kind of work, thinking about the regional forests, thinking about climate change. But I wanted to try and bring kind of a more like equity and inclusion approach to my science, like actually bring it into the science as opposed to it always being this other thing that I was working on. And I spent a lot of time thinking about it, and it seemed like, and reading of very smart people who have thought about it more than I have, and it seemed like the interface of plant ecophysiology and like environmental justice is a lot of urban tree biology. And so because there are disproportionate like places like lower income neighborhoods or neighborhoods of primarily BIPOC people typically don't have as much green space, and that has a lot of cascading negative effects on their mental health, on their physical health, on a lot of different things. And so I was Googling and I was looking, I found this really amazing paper written by Jonathan Thompson at Harvard Forest, where they mapped environmental justice neighborhoods across the Northeast. And there were like 8 key areas. And the way they defined environmental justice was an area of either a large majority of people of color or lower income, and then also lack of access to green spaces. And so they did this really cool map. And one of the main places that jumped out was Holyoke. And so I did some Googling, and I found that there's this amazing guy at Holyoke named Yoni Glogauer, who is the head of their conservation and sustainability department at the city level. And him and his team over the years commissioned this really amazing report called the Urban Forest Equity Plan. And so a lot of different cities have these kind of urban forest plans, like it's pretty common, but they are really unique and put centering equity in the middle of it. And it was this stunning report, just like trying to like map the trees, but then also like surveying residents, trying to figure out what's important to them and like where, just like all these different things. Super long document, it's very cool. So I read it, I reached out to him and I was like, I'd love to support you. Like, it seems like in the document, like it was kind of that one of the main conclusions at the end was, okay, we had the main conclusions are we don't have enough people, we don't have enough funding to like make the dreams come true in Holyoke. in regards to the forest equity plan. And I was like, okay, we got money and we got people, like, how can, what can we team up? And he was amazing and super receptive. And so we've just started working together. In the fall, I had my class. I taught an upper level class on climate change and plant biology and art. And my class went and helped him with the forest inventory. So like actually just going around Holyoke and mapping different trees and their shape or their diameter and their health and the species. And so it was great for them because they were learning about tree identification. And then it was great for him because he was getting some information for his tree inventory. And so we're trying this summer, we're going to have a couple of students working in Holyoke. And the question that we kind of co-produced together was, he's really interested in understanding how they just got like 90 more tree pits. So like the things in the sidewalk, they're just like these big things where you put the tree. And so he just got a lot of funding to make more of these. And so he's really curious about like which species does best in these situations, and like how much does like the size of the tree pit matter. And so we're going to have students studying those things by measuring like tree health through different tree physiology measurements over the summer, and like tree pit conditions. And then, okay, so that's one thing. And the other thing is I'm part of this new urban tree ecophysiology network that is an international group of ecophysiologists who are going to start doing like baseline measurements at all their different urban forests. So trying to get some understanding of like general trends or patterns, because we really are far behind in our understanding of the urban forest and like what are its stresses. Like some people are like, oh, they're definitely stressed for water, and other people, they're definitely not stressed for water. They have all this runoff. And so there's just all different types of situations. We need to like be doing more work around it. Like one of my favorite things we learned, that we had a conference for this network or workshop, and we had all these amazing speakers, and one of them was like just showing us all of these cool things. Like there's a tree that's like this big and that's like 100 years old, and there's a tree that is this big that's like 20 years old, and it totally depends on like where they're planted or what species they are. And so we're going to try and like do like a systematic survey of like stresses and health. And so I'm going to be doing that primarily in Holyoke, I think, like for the next few years.
That's gonna be so cool.
Yeah.
Oh my goodness.
I'm very excited.
So I had the pleasure of getting to preview your slides, but if you could show us the picture of the tree pit, because I think that's just so fascinating.
Yeah.
Like to me, that is just so crazy that you could expect any part of a large tree to fit in that.
So Yoni Glogauer, the person I was mentioning, this amazing urban forester in Holyoke, he like came and talked to my class. He's just so amazing, so excited to communicate and like get this. It's awesome. And one of my students was like, what's in a tree pit? Like, what's going on? Like, what's going on with the trees? And then the next day he was literally at a tree pit. He texted me a photo of it. And I sent to my class and I was like, well, this is the photo. And so this is a newly completely dug tree pit. It's like, you can see it's a couple feet deep. Like you can see a foot up there for scale. So, and then they're going to put a plant in it. But then if you look at a root system of like this is a pine tree, it's like, what? That's going 10 meters, 30 meters deep, super wide. So just kind of thinking about, like the unique stresses of the system. It's like, it's wild that anything can grow there. It's very, very impressive. And then you might already know this, but I didn't know this. I was at this, at the workshop for the urban tree ecophysiology, and I have never done urban biology before, so I didn't know. And I was just like, but can't a root just kind of like go like left or right? Like it's dirt. So I'm like, why can't it go in it? And they're like, absolutely not. Like it is super rare. And so sometimes you see it like in a sidewalk crack, like that's kind of like it found this like very specific area between the sidewalk and the soil. But to put cement down, this is where I'm really stepping outside of my range of knowledge, but I think this is true. To put cement down, they have to make the soil compacted to like 95% compaction. And it's like trees cannot function in that. And like there's also no water, there's also no nutrients, like it's really just dead, non-functioning soil. And so so they're like essentially bonsai plants. Like that's what you do when you put it in this situation. Like you're limiting its root growth so it can't grow that big. So that's why you get trees that are like 100 years and like this big. But yeah, thanks for reminding me about that. It's so crazy. Yeah.
That's crazy. Oh my gosh. So you touched a little bit about this when you talked about your class, but you're incorporating art into a lot of your lab, to your classes. And we're going to talk about this a lot more in depth later, but just to kind of ask you right off the bat, why do you think that it's so important to incorporate art into your science and your lab culture?
What a question. Okay. You can send me these questions ahead of time, and this one already stumped me. Yes. So yeah, so I'm incorporating art into my classes, and then I make art, and my like technicians in my lab make art. And we show the art, and I think there's a bunch of reasons, but first and foremost, I love art. So I went to a creative and performing arts high school in Philadelphia, and I was a professional tap dancer before I joined this realm of the world. And I'm also a poet, and so the art is a very, very big part of my life. For most of my life, I identified primarily as an artist, and then I ended up being a scientist. I'm still like dealing with the identity conflict. But so first and foremost, I love art. But you know, the justifiable reasons why we're putting money into it, why the NSF is funding, you know, there's money going into it, is because, I mean, okay, so I'll try and break it down. So like one is it increases, I mean, there's studies that show that bringing kind of your culturally relevant artistic practices into a classroom or into the lab, it makes it more accessible. It makes it more inclusive for people who are traditionally underrepresented in STEM spaces. So that's one reason. Another reason is there's also studies that show that it changes your mind. Like it really does. Like when you integrate art, it allows you to think more creatively, which makes sense. But like there's, you know, science so sure it allows you to think more creatively, and it allows you to have more like big picture thinking when you're thinking about your projects. And then another reason why I incorporate into classes is because it's really, well, oh my God, okay, it's really a good, powerful tool for community building. So I have art in both of my classes that I've taught this at Smith. It's like pretty much the only assignment they have are making art projects. There's a few other ones, but those are the biggest ones. And they then present them to the class, and we have workshops in class about their art pieces, and they're all tied to things we're learning about, and that like physiology or ecophysiology. And it allows them to really, like, they're learning, like they're really processing that. It's like a fun way for them to meditate on a process for a long time. And so then alongside of the art projects, they have a write-up where they have to tie either like the product or the process or something about the art project to something we learned in class, and then they have to describe it. And these things are so stunning. Like it's absolutely amazing what they do. Like I could give a billion examples, but the first one that comes to mind is my student Emma King last semester. She made a infinity mirror. So it's like, it's all different forms. There's some people doing like collages and paintings and writing and poetry and singing and all these different things. But she liked to make 3D sculptures. And so she built an infinity mirror with moss inside of it. So you could like look at it and it looked like moss forever. What I still don't know how she made it. It was very impressive. And I was like, Emma, this is really cool, but like, how does this relate to what we're learning? And she was like, well, like in class, we learned that like, if you take findings from one paper and extends them to the reality, it like gives you a false sense of reality. And like, so she really started thinking about like our relationship to scientific literature, like through this project. And it was just really stunning. I had another student who we were learning about the process of transpiration to water coming out of the stomata. And she, this was amazing. This is Mark Foma, who's my TA this semester. She was like, okay, professor, I'm going to collect water from the leaves. Like I'm going to put a little bag and collect water transpiring, and then I'm going to use that to watercolor a picture of a tree. And I was like, it's impossible. I was like, it's a really cool, thoughtful, creative idea, but like, there's literally no way you could do that. And then she totally did it. She totally proved it wrong. And she was like, yeah, it took like a couple weeks. I had to figure out exactly when to put the bag and when to take the bag and like to make sure that it wasn't all being evaporated. And then she made this stunning, like 3x6 painting of a tree. It was amazing. So it really gets like, and so she learned a lot about the process of transpiration through that project. And so, yeah, I could go on for a really long time, but so those are some of the main reasons why I incorporate art.
That is amazing. That's so cool. All right, that brings us to the end of vignette 1. So I would like to now open it up to our audience and see if anybody else has any questions out there. Yes, in a blue shirt.
Regarding the xylem, well,
Oh, yes.
Tree, I guess. How do they form? Is it the fetal layer of cells rapidly dividing in between them? And
That's 100% what's happening. Yeah. The vascular cambium.
Ah.
So it's exactly what you think. So there's a meristematic cell region between the xylem and the phloem. And see, yeah, there's not a photo over here, but It's exactly that. It's rapidly dividing, and every year it makes some more cells. And what happens is, if you look at like the cross section of a tree, you see rings. We all know this. And the rings are a year's worth of xylem. And so that's what makes a tree grow. But then the phloem gets crushed every year. So it's not as robust. It doesn't have the same secondary thickenings of xylem. So it just gets crushed into the bark. And so there's like an area of crushed xylem that should be equal in size to the xylem, to a stronger.
And does the xylem phloem extend into the roots?
Yes, it does. I just had my students build some roots out of clay. That's what I hated report, but I think it was worth it. But yes, they had the xylem, the primary xylem and primary phloem form like 0.5 millimeters away from the root tip. And so if you think about, there's like a shoot apical meristem, the thing on the top of the tree, and then there's a root apical meristem, and that's where they kind of grow. And both of them are making xylem and phloem. Yeah, cool question.
Yes. Okay.
Me?
Yes, you.
So we're in a neighborhood question where somebody cut down 25 trees on a lot that floods all the time. So when you said 100 gallons in a big one,
Yeah.
they weren't all huge. Okay. But is there some rule of thumb, or is there some way for us to kind of try and estimate the amount of water that those trees were capable, or similar trees would be capable of taking out of the soil
Yeah,
on
I
a
mean,
daily basis?
yeah, you definitely could. It would take, there's no, I guess there's not, my rule of thumb is if it's a huge tree, it's 100 gallons, if it's smaller, it's less. But, I mean, you could try and dig into like literature on it. Like there definitely are measurements of how much water is coming out of, and the units are annoying because the units are typically on a per leaf basis. So you would want to know like how many leaves there were, and then you know 2% and like 30% of photosynthesis products going into root exudates for trees. I think it's for trees. There's, like I said, there's not a lot of research on it, but it's a non-trivial chunk of carbon going underground to feed these systems. Hope that's a good answer. Yes.
Can you tell us something about the drought situation in the west and how that's affecting all the trees out there?
Yeah, so I mean, I could, yeah, so it's not great. I mean, it's really not great. It's really sad. My students were crying in class about it last semester because we watch all these horror, like it's horrible. Like watching the videos is really like horrible. I guess the biggest thing I would say is it's typically a bunch of different things. So it's the drought first. So the drought weakens the tree because it minimizes the amount of photosynthesis you can do. So it's already kind of sick. And then that's right when the bark beetles will hit. And then the bark beetles will take advantage of these trees that are already stressed. And then the fire will hit it, and then it will burn all of it. So it's like a bunch of different stressors coming in, but drought kind of weakens them initially, because I guess one of the papers I read about recently was about when you have a drought, you have less ability to make these secondary defense compounds and resin that protects you from the bugs or the beetles. I don't know much about insects. But so they have this like actual physical mechanism where they're like exuding resin and they're exuding these defense compounds. And if there's less photosynthesis, they do that less. And if there's less access to water, they do that less. And so they have less protectance against the bark beetles. But yeah.
The bark beetles seem to be doing a very good job with a lot of the evergreens.
Yeah, they are heartbreaking.
Does anybody else have any questions? Yeah.
I'm just curious about the soil pits. Are there, and thinking kind of from the art lens, I know funding is probably the limiting factor, but are there other designs? Are like,
Yeah.
Can you go deeper? Can you? I'm
Yes.
curious what else about.
So at the workshop, I learned a lot about soil and the Urban Tree Ecophysiology Network. They had different like engineering groups come, and they're all these, there's like what they call tree tubes was the one we learned about, where they kind of like, because part of the reason, like I said, is the compaction of the soil. And so if you can find ways to not compact the soil, then you can have more area for the roots to explore. So that's the main thing, is people are trying to find ways to not compact the soil, but still be able to have a walkable surface above it. And so they're doing all different types of things with like things that look like stilts, you know, all these different, they have a lot of different ways of doing it. And it's definitely working in some areas, like cities that are really taking their green space, like are putting funding towards it, they can fund those types of projects, which is very cool.
Can I speak to that? I'm the chair of the tree committee in Amherst.
Oh, awesome.
So, yeah, it's totally right, you put the trees in these pits and they're just too small. Trees will grow roots, if there's grass nearby, they'll grow roots on the sidewalk and then take advantage of all the nutrients there. But, one thing that, is used, it's very expensive, but it's called structural soil.
Mm.
It's a type of soil that doesn't compact, it's got a lot of larger chunks of things in it. And, that really helps to keep the roots alive. But the main thing is just try and make the tree pits as big as possible. And we did a few downtown where we actually tore out the concrete from the sidewalk so we could enlarge the tree pits. The tree manager was saying, how come the downtown trees are dying? Well, it's not enough space. He said, why don't we make more space?
Yeah.
That's one solution, but it's challenging.
Yeah, that's awesome. Thank you for that.
Yeah, that's super helpful. We're gonna have to keep moving, but amazing questions. Thank you all so much. Okay, so vignette 2. Now that we've kind of had a primer on trees, we're gonna get a little bit more in depth into your research.
Right.
Okay, so first off, your lab is called the PLACE Lab. Can you break down that acronym for us?
Yes. Plant physiology, so I had to stretch it a little bit. Plant physiology, art, and community engagement. So those are the 3 pillars of the lab. And I put them into the name because I was like, I want to hold myself accountable to these principles. Like, I don't want to just like fall into one because that's like what's expected in an academic setting. And so we do a lot of plant physiology. We do phloem ecophysiology, like I talked about. We do the northeastern trees response to drought and cold, and we do the urban tree ecophysiology. Yeah, is that, yeah, so those are the big 3 components. And so I guess for art, we talked a little bit about, we have art integration in classes. I have a policy in the lab where anyone who works in my lab can spend 20% of their paid time making art projects related to what we're learning about. And so it could be an art project, it could be a community outreach project, or like a community art project, but also they don't have to, it's just if they want to. And then there's community engagement, which has kind of like 2 approaches. One is like engaging with communities outside of Smith, and then also supporting communities that are historically excluded from STEM spaces. And so those are kind of like the pillars.
That is so cool. That's such a unique approach. I haven't seen it before, and I think that's super interesting. We want to focus for right now on your work with trees. So it seems like you work on many different branches, pun intended, of tree ecophysiology. So how would you personally describe your science?
Okay, so how would I describe my science? I study how trees are dealing with the environment. That's like very much what I define, like is kind of the general theme between all of the different projects. And right now it looks like these 3, I have the 3 different projects, the phloem project, the urban project, and then the Northeast region project. And they're all focused on slightly different physiological questions, but they're all very much related to like, how is the tree handling what's going on outside?
Sure. So one aspect of that is climate change. How do you measure climate change? And how do you correlate climate change to some of the changes in physiology that you're seeing in these trees?
That's a great question. So I do not quantify climate change. There are amazing climate scientists out there doing this work through all different types of climate modeling and climate measuring. And so I spend the majority of my time doing the plant work, and then I just rely on papers from other fields to kind of inform that part. So like in the Northeast, a paper came out like 1 year ago, maybe a little bit more than that, that showed that the northeastern region along the ocean is warming faster than any other part in the eastern US. And that includes where we are. And the reasoning for that has to do with the ocean stuff. I tried to go into it for classes. Generally, it's because of currents and the way everything is warming. So I would like rely on a paper like that that's showing me that the eastern region is warming super fast. And then there's people who do all types of climate modeling, looking at drought modeling specifically. And there's papers that have shown that droughts are extreme events are likely to happen more and more in the Northeast. It's tricky in the Northeast because it's really stumping a lot of, I think, or it's stumping me. I'm reading all these papers. But I feel like some people are saying like, we're gonna have more precipitation, but it's mainly gonna be in the winter. And then there's also gonna be extreme events, so there could be more and more drought during the summer, even though we have the precipitation. So like, you can't really look at annual, so there's all these things you can get into about being really specific. But there was a paper that came out a few years ago that showed that the Northeast in all different climate change scenarios, so from, you know, business as usual to like super low emissions, all the way up to like hellscapes. All of those situations, most of those situations predict more early summer droughts in the Northeast, and that is the timing that's really important for trees. So in terms of quantifying climate change, they do it. Those brilliant people who are masters of these models. And I like look to things like the IPCC report, the Intergovernmental Panel on Climate Change. They do these amazing reports every like 6 years, where they aggregate everything we know about climate science, and then I pull from that to inform like what questions I'll be asking.
Very interesting. So we looked at your study on water dynamics in oak trees. Could you just talk briefly a little bit about this study and how it relates to the phloem ecophysiology?
Yeah. So that was my first study for my dissertation. And I loved it. It was really fun. I got to use that fun bucket truck. And so we were thinking about daily stress. So okay. So we were thinking about diurnal patterns or daily patterns of carbon movement in trees. And I ultimately was motivated by thinking about droughts, but it's really hard to drought a huge tree. And people are doing it. And hopefully we're going to maybe do it at McLeish Field Station, which I'm really excited about. We put this huge tarp thing out. Anyway, so it's pretty hard to do that. And so my advisor, Missy Holbrook, who's just like the bee's knees and very, very, very smart, she was like, okay, let's think about daily water stress, because every day in the middle of the day, there's like a mini drought for a tree because the stomata are open, water's gushing out of it. And so it gets water stressed every day. And so we wanted to see how that daily water stress influenced carbon movement in plants. And so, and we thought based on the literature, but like I said, there's not a lot of literature on phloem biology, but based on a few amazing papers that were out there, we thought that that daily water stress would be enough to stop the phloem. And we thought that the phloem would be working at night when the plant is most hydrated because the stomata are closed, it's filling back up with water. So we thought that's what happened, but of course we found the complete opposite. We found that in the middle of the day, all of the garbage moving everywhere. It's like this amazing thing, despite this water stress. So then that kind of motivated the rest of my dissertation, which was kind of trying to be like, okay, well, how is this happening? Like based on our understanding of the phloem, based on our understanding of the sugar concentrations in the phloem, this should not physically be possible. And so then we developed it. So after that, we then ended up developing another method to measure sugars in the phloem using this thing called Raman spectroscopy, which has nothing to do with the noodles. And but so then we used a new tool to try and look at the sugars, and we found that it was much more sugary than we thought. So the data in the literature suggested that the phloem had about the sugar concentration of a Coca-Cola, about 500 millimolar. But we found it was 3 times that in the leaves of trees. These things are sugary, they're tiny and they're sugary, they're like pixie sticks. I just had a pixie stick recently. And yeah, they're just like that. So anyway, yeah. So then we had this kind of series of papers where we first were looking at the daily stress. We found that's not enough. And then we found out it's because they're super sugary. And then we're like, okay, we got to super drought one of these things. We got to figure out what's going on in the drought, because that's what's important and interesting for thinking about carbon cycling. Then COVID happened. And so we ended up using some little plants. I don't think I have a photo of it, but Ricinus communis or castor beans. And so we used some of those that are like this big in a growth chamber, and we droughted the crap out of them. And we found out that the phloem is just amazing. It can handle it. Not only can it handle it, it doubles its sugar concentration during the drought in order to handle it. So it's really dynamic. So we found out it was dynamic and it can handle stress, and it's great for trees.
Wow.
There's still a lot more work to be done. That was like one species, but so oh yeah, great. Oh yeah, they're the so yeah, they're the castor beans. And yeah, so we found out that they're really dynamic, which is great, but now we have some more funding to try and start looking at bigger trees, droughting bigger trees, and looking across species. So that's like the next steps.
That's super, super interesting. Okay, well, that actually brings us to the end of vignette 2. So we'll open it back up to our audience if anybody's got any questions. Yes, we'll go here and then back up.
What kind of measurements, what kind of instruments do you use in order to know
Yeah.
what's going up and down?
It's a great question. And it's fun, and I'm happy you brought it up because this is a huge part of plant physiology. We're a very method-based field. So we're always trying to develop new methods and because, yeah, anyway, so here's one. It's a box. So this is the base of it, but then it has this like head on it that you clamp onto a leaf. This thing costs like $80,000. Not exaggerating. I'm trying to buy one right now. And not good, yeah. Anyway, so it clamps onto a leaf and then it measures gas exchange. So it has a gas analyzer inside of it. And so it can measure how much CO2 is being taken up and how much water is coming out from a leaf, right? Just like clamping onto it. So that's like a primary method. There's, you see me here, this is me pumping a bicycle pump. We did some remote field work in Chile to look at drought stress on these amazing plants. And that's to use, it's the same thing, you can't quite see it, but it's here. And that's called a pressure chamber. And the way that
There it is.
oh yeah, perfect, okay, so you see, this is a little nitrogen gas tank. When we were in Chile, I was the gas tank and I was pressurizing it by hand, but usually you can use compressed air. And you put a little leaf in this thing, chamber, and you compress it with a lot of gas. And then when you're looking at it with a little hand lens, and then you pressurize it until water starts coming out of its petioles, little stem only. And so once you see that happen, that's how you know how stressed it is. And so if you need a lot of pressure, it's super stressed. And if it comes out immediately, it's very hydrated. So that's another tool we use. We also use a thing called an osmometer. That's not as cool looking. It's just like this device that tells you how many solutes. It actually was built for blood analysis. And it tells you how many solutes are in the leaves. We also do a lot of like wet lab chemistry, so a lot of pipetting to measure sugars and starches and like I mentioned, Raman spectroscopy. Yeah, so it's a lot of fun, a lot of tools. Yeah.
And all that sugar comes from the little stem that connects it up to the bract.
Yeah, so all the sugar is made in the leaf and then it goes through that tiny little,
tiny
petiole, goes through the petiole and it goes all over the body. Yeah, it's amazing.
Amazing.
Yeah.
Going back to what you said earlier, and it's hypothetically, the 100 gallons of water that the tree is looking for in a forest, you have a whole bunch of trees all looking for that same water.
Yeah.
But in a city, there's no competition.
Yep.
But the water isn't in the ground, it's run off on pavement into storm drains.
Yeah.
Which is one reason why the plants are strong enough to break into a storm drain. Cut them out.
Yeah.
So, which is it for those urban trees?
Yeah.
They have an abundance of water or?
It's a great question. And it's something that, so we're gonna try and investigate. There's reasons to believe that they're quite drought stressed because of the limited area they have, but you're right, like if the topography of the city is funneling it into that little pit, it could be really happy. If it's like just this little pool for them to be living in. I mean, they wouldn't want to be in a pool, but you know what I mean. But yeah, so it totally depends on the topography. There's all these, I mean, you probably heard about it. There's all these really cool new, I mean, not that new, but infrastructure approaches like things called rain gardens, where they like make the topography so the water goes into this area and they have a bunch of plants in it to take it out so it doesn't go into the sewer system. And so people are starting to try to play with that. But generally, I think the general assumption right now is that they're usually pretty stressed. Yeah, it's a great question.
And what is the road salt?
Oh,
Yeah, I mean,
mess.
obviously that's not,
yeah. it's
is it
horrible.
diluted enough because there's snow as well?
This is a good question. Generally, we think of it as a stressor on the plants because, I mean, so if we think about things like osmosis and diffusion, if you have a lot of salt in an area, that just makes it thermodynamically drier. So it's drying what's already pretty dry. So it's not great. And then you got to think about the chemical effects of it. And so I haven't seen a bunch of studies, but to the science world's defense, I haven't been looking so much. So I wonder, there's probably some really cool research on it, but generally I think it's not.
Because when you see the trees in like New York City, I mean, they
Yeah.
have amazing trees on the sidewalks, giant pear trees and things. So, and obviously they're getting a lot of salt.
Yeah.
So I guess, I mean, even though it's a stressor, they can seem to survive.
Yeah, and so they plant certain species that they know are pretty resilient. So I think that's a key to it, but there's still a lot of work to be done to identify those species, like who is kind of more salt tolerant, because you can do things to deal with salt. And it changes species by species. But yeah, it's a great question.
I can add that, it's also the salt spray. So along faster roads, the spray comes from the cars, landing right on the leaves or right on the stems. If they're using a stem.
Yeah, because you
That's-
see the brown trees at quite a height.
Yeah,
the
that's
spray.
worse than the actual salt.
Yes.
I have a linguistic question. When you say the urban forest,
Yes.
okay, when I go into a forest, if I only see one tree, I don't consider it a forest.
Yeah.
But what makes the urban forest forest?
This is a great question. So I'm probably not the best person to speak about it because I haven't thought as deeply as some of the like urban tree biologist scholars. But there is kind of a movement that I've been seeing in urban ecology to try and start really viewing this thing, like what is an ecosystem, what is a forest, like how do humans relate to these things? And it's like, of course, yes, 100%, it's super different than like forest. I completely agree. But we're trying to think more about the ecology of these systems. So thinking about like, how do bugs depend on these? How do birds depend on these things? Like, how do all like the ecosystem functioning? So there's kind of a movement to try and think about not just as like a street tree, but like have a broader understanding of it as an urban forest. That's my very short hand-wavy answer, but there's definitely people who are thinking really deeply about this, who are kind of pushing for that more
So
perspective.
there's that whole element of how trees relate to other trees.
Mhm.
That's completely absent.
Yeah, so it is, unless you're in something like a city park. So like there are situations where they can, and I just was teaching about this. This is one of my favorite things I've been learning about. Trees can also communicate through the air through volatile organic compounds. And so they essentially send these signals to other trees through the air. And so there can be communication above ground between different street trees and stuff. So it is, but you're totally right that like it's less, they don't have any of the underground situation, which they're totally related, totally dependent on in a lot of cities, or not dependent on, but it's important for them. But there still are, they have traces of communication.
One question, do you think that in the urban forest that the volatile communication might be stronger than in the?
You could imagine, yeah, you could imagine, I'm not sure, well, it's tricky because I don't know if it's like so if we think about it in terms of acclimation versus adaptation, so for non-evolutionary biologists, so acclimation is kind of like what an individual can change about itself, whereas adaptation is like this genetic thing, so it goes on over the years. And so I think in terms of the trees, there's definitely a lot of adaptation happening in the urban forest, but in regards to like bugs and other things that have really short lifespans. But for trees, I think it's less so. But if they have the ability to acclimate, I could imagine they do, and that they are trying to communicate more that way. I can imagine. I don't know that it's ever been tested, but it's a great question.
But there's more competition. There's a lot of loud noises around to interrupt the communication.
Oh, yes. Well, zooming cars.
All right, I think with that, we are going to move into our final vignette. And this is something I'm really excited to talk to you about. So you've mentioned that you have just started. You're getting your lab kind of up and running. Could you tell us a little bit about what it's like to start a lab? Like what kind of logistics do you need to think about? What do you wish that you would have known before you got started?
Maybe I should defer to my partner on this one, but I'll talk about the experience for both of us. It's been very time intensive. Yeah, so I was lucky to walk into a lab that was a previous plant physiologist lab. So they had a lot of the basics. They had the $70,000 machine that measures these things. They have centrifuges, they have pipettes, they have a lot. So I didn't have to go through the rigmarole of getting all of these like basic lab supplies. They were there already. So that was a huge, great thing. in terms of, I mean, the truth is that I'm still very much getting it set up. So this year, Smith was really generous and I have a really great teaching load. I just teach one class with a lab each semester. But somehow that managed to take 90% of my time. So I spent a lot of time coming up with the course material. And I'm slowly building the lab. I got really lucky. I have a collaborator at Holyoke Community College named Sage Fernetovich, who's their botany professor. And she was like, I have an HCC student who's coming to Smith who you need to snatch up immediately to be like your technician. And I was like, okay, great, thanks. I know you were talking to, and he has been a complete blessing. He's like super organized. He ran the greenhouse at HCC for years. He's like super skilled. He's a non-traditional student. So he's like, he knows what he's doing. So he's older. And so he has been almost like an equal in setting up the lab. He's been like, Professor Gersony, you gotta do this. And I'm like, okay. And I go in order for so that's been really nice having a team. I do much better working with others. I really like collaborating, and I am much more productive and happy in that situation. So I'm really happy to have him. He's been awesome. And this year I'm hiring a postdoc who will be a full-time researcher in the lab, and then also a full-time technician. So in the next year, it's gonna triple in size, the lab. So that'll be really great in terms of getting research up and going. But generally that part hasn't been too intimidating yet, but that's because I haven't been doing that much research yet. But I think building the connections is like where I put a lot of the energy, like trying to find people in the communities who like I can engage with and think about trees with. Yeah, I don't know how useful that helps.
No, totally, that's such a totally unique perspective. In terms of building community, as you're starting to think about who to talk to and who to reach out, like, what are some of your thoughts on how do you even build community?
Yeah, that's a great question. So I think there's a few different realms of, I mean, there's a billion realms of community, but so the ones that I like, I guess I'll speak to. So one is there's a plant physiology community. It's super international. There's probably like 20 phloem physiologists in the world. Like it's not a very commonly researched thing, and I pretty much know most of them because there's so few of us, and so we're all reading each other's papers and stuff. And so we have a meeting group that meets every couple weeks on Zoom, and so we all get together and like read papers together and keep each other up on like what's going on in the labs. And that's really amazing. That's run by Jessica Savage at University of Minnesota Duluth, who's another really big mentor of mine. And I love that she does it. Like, I just think it's been really great to have that community because a lot of us are at smaller schools and like don't have other flow biologists around us. So leaning on that has been really nice. And then there's also the greater ecophysiology community. So there's, a few conferences a year that are like 100 people, and we all kind of know each other, and we all go all over the world. And we do like, I just met them all in Georgia, and then we're going to meet them all in Maine, then we're going to meet them in Bordeaux, so that's really great. So there's a lot of community outside of the local area. And then in terms of, so that's like the research, like my physiology community. And then I'm working on trying to build connections with different partners of the forest. So like people who think about the forest differently. So like when I got here, I met with a few people from Arcadia, and I met with Joni, who ended up being one of my good collaborators now, an amazing person. And then at Smith, there's a pretty great plant community at Smith because we have the botanic garden. And so I'm very lucky in that regard. There's Gabby Immerman is kind of like the main educator at the botanic garden, and she is a master educator. Like I have so much to learn from her. And so that's been really helpful, like talking to her about teaching. And then Jesse Bellemare is the other plant biology professor at Smith, who's just so cool. Like he's so giving of his time, like he's so supportive, but he's also like so calm, which I have a lot of energy, so we balance each other really well, and he helps me really think through things, and he's just brilliant. So I've been building that kind of community, and then I've been reaching out to like local foresters and trying to build a network of people to kind of consult to make sure that research is relevant to the people on the ground. And I'm very much in the beginning stages of that, but I've been having some preliminary meetings with people and starting to try. I just cold email everyone. That's my general approach. Sometimes usually it works. People are usually happy to chat, which I really like and appreciate. But yeah,
That's wonderful. So Another kind of aspect of community in some ways is diversity, equity, and inclusion. Could you tell us a little bit what that looks like in practice and what kinds of approaches you're trying to take for your own lab?
Yeah, okay, so, this is, I think, half of, is there another, I don't know, maybe the transfer, yeah, okay. So I tried to list the different things that I thought DEI meant to me and like what it looks like in practice. And there were a bunch of them. It's something I really, don't want to be doing the work unless I'm doing it in this context and like, unless I'm challenging myself to be better in this regard. And so the first thing that I kind of say at the beginning of my classes and to myself when I go to bed at night, no, but like generally when I'm thinking about my priorities is I'm trying to reimagine academia by identifying with shifting white supremacist practices. And so I went to this really amazing workshop, like right when I started at Smith, called Academics for Black Lives. And it was so amazing. I learned so much. It was 2 weeks long. It was like unlimited content of amazing Black scholars. And so they kind of like through that, I like was learning more about, okay, what does white supremacy look like in academia? How can I identify and how can I, now that I do have the power in a classroom, in a lab, shift those practices? And so I'm trying, I mean, it's very much a work in progress as this work always is. But so I've been trying to like name the things in the beginning, like in the syllabus, I have practices named that are things we're trying to shift or change. And so the ones that I focus on are, these are kind of typically considered white supremacy practices like perfectionism, urgency, product over process, individualism, and quantity over quality. And so those are things I'm trying to be mindful of in the classroom. And I do that through a bunch of different ways. The main one is having community building be at the center of the class. So I spend the first week just community building. Like I don't even get into content because I want, because there's research that shows like if people are in a positive learning environment, they're gonna learn more and they're gonna learn better and they're gonna remember things more. So that's one reason, but also just because it's great. It's a really nice community when you go into it every day. So community building is very, very important. And I've told my students time and time again, like, I don't care if you didn't do the work. Like, I don't care if you didn't do the reading, like still come to class. Like you are wanted here, you are respected here and you're cared for and like, so that's one thing I think a lot about. I'm also doing a thing called ungrading, which is kind of this new movement in pedagogy to kind of try and take the grades out of the classroom. We have Hampshire, so you know, guys, maybe people were familiar with Hampshire. They're at the forefront of this. But kind of taking away numeric grades and giving just feedback and having one-on-one meetings with students to talk about progress. So I've been doing that in both my classes and it's been going really well, I would say. Like I said, I'm incorporating art, which I think has a DEI perspective if you think about inclusion. I'm incorporating traditional ecological knowledge into the classroom. So I do that not through, well, I guess the way that I do it is we read a lot of papers by Indigenous scholars in terms of both like peer-reviewed literature, but also non-peer reviewed literature, just about like how to think about TEK and how to integrate it into the classroom. We were thrilled when Robin Wall Kimmerer came to Smith this semester. And then I have really like, along with the ungrading, there's kind of like, no, some people might be thinking I'm going off the deep end, but there's no real deadlines or there are deadlines, but they're suggested and there's really flexible attendance policy. The only policy is that like, I trust that you're going to come when you're healthy, that be it mentally or physically. I also think a lot about spreading the wealth in terms of research opportunities. There's a bunch of really amazing students at Smith who want research opportunities, but we can only give so many because we're only have the capacity to do that. So I really try and select students who haven't had research experience or who are typically underrepresented in research spaces. I'm also on the college's Inclusion Council, which is kind of this group that gets together. There's like one person from the sciences, one person from the humanities, one person from the arts, and we meet with the what is he called? The vice president of equity and inclusion at Smith once a month, and we kind of just like talk about what's going on at Smith, what are we hearing on the ground. And then I'm also on the bio department DEI committee. Yeah, centering environmental justice in the syllabus. This is something that I did this semester, and I'm really happy I did it. At first I thought I was way over my head. I still feel that way, but I don't regret it. So I tried to because when you teach plant physiology, you typically teach it in an environmental plant physiology context. And I tried to kind of add one more word to that and have it be an intersectional environmental plant physiology context. So we were learning about like this kind of urban tree stuff that we've been talking about. We also learned a lot about food justice and the agricultural system, and we also learned about plant physiology in the context of nature-based climate solutions for thinking about climate change as a environmental justice issue. Bringing our whole selves into STEM spaces. So like we do a lot of journaling and sharing, and that goes along with building community. And there's a bunch of other things that's for us to start. Yeah.
Do you have any other suggestions for people that are looking to get either more involved in the DEI sphere or make more of a difference in the DEI sphere?
I mean, for what it's worth, I suggest like looking for groups that are already existing. Like I think in a lot of spaces there are, like at Smith, there already was the Inclusion Council, and the department had a DEI committee. So I think looking for those places, and then just it's from my experience, they're happy to have new people. Like I kind of couldn't believe it. I got to Smith and I met with when I got there, I met with the vice president of equity and inclusion because I just wanted to know what was going on at Smith. And then like the week later, he's like, do you want to be on the Inclusion Council? And I'm like, out of all the faculty in the sciences. But so he was very happy to have that. So I think that just like reaching out. And I think like trying to really learn about white supremacy and like learn about its impacts on academia is like where to start. I think that's kind of, I'm still there and I think I'll always be there, right? Like I'll always keep learning more and more about it, but then trying to figure out where you have power. So like I'm in a situation where I have power in the classroom, I have power in the lab, I have power at Smith because I'm a faculty. And so trying to figure out what I can do in those realms. Like I'll get super depressed if I think about white supremacy at large, because like I'm not the president, you know, like I'm not a congressman or somebody who can like make these huge shifts of wealth. But I do have power in the community that I'm in. And so I'm trying to use that and be really mindful of that and like dedicate a non-trivial amount of my time to the work.
That's awesome. That's super helpful. Another thing, we've touched on this, but now I'd like to get a little more in depth of is the prioritization of art within your work and in your students' work. How do you kind of communicate the importance of art to your students?
Oh, I don't even need, they get it. They're so on board. They're more on board than they're. So when I got to Smith, they said I could teach an upper level course in my area of expertise. That was the framing. And I was like, like, what is my area? Because I teach an intermediate level plant physiology, course on plant physiology, which I'm doing this semester. And so I asked them at first, I was like, could I teach a course integrating art and science and climate change and plant biology and all these things that I care about? And at first they were like, sure. Like, what? So then I wrote a proposal. I wrote like 5-page proposal saying like all the reasons why it's important. And I like went into the literature and it's there, right? There's so much science behind STEAM education. So integrating art into STEM spaces. And so I just was like kind of pulling from that and like thinking about climate change is such an interdisciplinary issue. Like we need interdisciplinary thinking in order to deal with it. And I really believe that. And then they were so on board. Like I have so much support from my department and the students, they're beating down the door. They want to be doing this. Like I mean, I think especially like at a liberal arts institution, but also I know it's probably biased sampling, but most of my best friends who are on plant biology are artists. All of them have their own practices outside of the classroom, be it like quilting or dance or writing. Like there's everyone, like most people are doing something, painting. There's a lot of beautiful painters. And so I feel like that I just felt strongly that we should try and find creative ways to integrate these things. And science can only do so much, right? Like we have the IPCC, we've known about climate change for what, 7 decades, 8, not 10, a really long time. And that's clearly not doing it. So this idea that like, we need to take multiple approaches. We need to show the science, but we also need to communicate it. We also need to express it artistically. We also need to be in policy. Like, I really believe in all of that. And yeah, so that's, I think, part of the reason that it was like communicating its importance. Yeah.
All right, I have one final question for you before I bring it out to our audience. So it was recently Earth Day.
Yes.
Could you talk a little bit about what Earth Day means to you as
Yeah.
a ecophysiologist?
Yeah, so it's very personal. So I am a scientist because of this man. His name is Dr. Richard Moon, and he was my high school science teacher. And a very like personal mentor, like he mentored me through all of college and into grad school, and just he would like be reading all my essays and like talking to me about my projects, and like he just was like a real father figure to me. He passed away a few years ago. But he's the reason I'm a scientist, and until he died, every year he would reach out to this like group of friends, like we had this little community, and every year he would remind us it was Earth Day. He was very sassy and sarcastic, and he's like, "What are you doing? Yeah, you gotta do something." And I just, yeah, it's so, it really, I mean, he affected me so much, I became a scientist for it. And whenever I think of Earth Day, I think of him and how much he's influenced me. And I was in a lab meeting last year on Earth Day. This Earth Day, I was at the Northeast Natural History Conference, which seemed very appropriate. But last year I was in a lab meeting, and I remember my PI, who's this amazing researcher at University of New Hampshire named Scott Hollinger. He was like, "Yeah, every day is Earth Day for plant biologists." He's like, "We're dedicating our lives to the earth," and like that made me feel really good. And it's like, I think, you know, I do treat Earth Days special, like I do, especially because I think of Dr. Moon and his influence on me. But yeah, I try and carry Earth Day in my heart every year, and that's part of the reason I dedicated my life to the work. So yeah.
That's amazing. That's amazing. All right, we've got just a little time for a few final questions from the audience, if anybody's got anything they'd like to ask. Yes.
Stop tap dance.
Great question. No, so before I did my PhD, I was in a professional company in New York City for 4 or 5 years. And then when I went to Boston for my PhD, I was in another company. And then it was too hard to do during my PhD to keep up with the rehearsals. So I kind of bowed out of that, and then COVID hit. And then I got to Smith, and I've been trying to figure out my relationship to the art form, because it's tricky because it is a Black art form. It was created by amazing Black tap dancers, and trying to understand my relationship to that as a white person, and like how I can honor that, and yeah, just honor that and not do harm. And so I think a lot of where I am right now is I think I'm going to start teaching. I think I'm going to start teaching tap either at like a local community center. There's a couple places in Northampton or Springfield, but also I might start teaching it at Smith. I might teach it at tap course there because they don't have a tap teacher right now. And so I've been in talks with the dance department.
That's so exciting.
Is there any town or city, talking about urban plans, that have a reputable reputation in terms of having done well with
Yeah.
their planning?
That's a great question. So I was just in, okay, so I'll start non-local, then I'll go local. I was just in Athens, Georgia, at the University of Georgia. And they boast the highest tree. There's some, I mean, it depends, but no matter what the metric is, they're generally at the top. They have like 55% tree cover if you're looking from bird's eye view. Yeah, it's a lot. But so from Yoni, I'll quote Yoni because he's very much at the forefront of this. And he said Cambridge, Massachusetts has a lot of funding, and also Northampton. He said Northampton is a really kind of a model for him in Holyoke. So yeah, in the Northeast, definitely.
Yeah.
I don't think this is, I don't know, maybe it's not as relevant of a question, but just thinking of like urban canopies, is there work to look at not only what trees do well in urban forests, but also if there are food producing trees that can do well, like, I don't know.
Yeah, that's a great question. So just today in class, we were talking about urban farming and what that looks like. And I am definitely not an expert by any means, because it kind of brings in the agricultural side of things, but there's definitely people doing this work and trying to figure out how they can create food in urban settings. And not only like it's really inspiring. We had a great conversation in class today that there's a lot of community-based urban gardens, like Philadelphia has a lot of really cool ones where they're kind of just like community members taking over vacant lots, and it's non-trivial the amount of food that these places are producing. So it's definitely something that's happening that I want to learn more about. I don't know a lot about.
There's a group in the valley that does that. There's actually a little food forest spot in Northampton. You might not have seen it yet. but.
What's the root?
Help yourself,
Cool.
yeah. I'll send you the link.
Yeah.
Do you have any more questions for Jess? Okay, well, with that, thank you so much. And thank you all so much for joining us. If you are not a part of our email listserv, we'd love to have you be part of it. That's where we send out a lot of information about our next and upcoming events. You can also follow us on social media, on Facebook or Instagram or Twitter. Our next event is actually going to be an all in Spanish cafe with Dr. Patty Brennan from Mount Holyoke. You can check out our website. And again, if you haven't already, you can fill out our sign-in sheet and we can continue to bring you cafes on the topics that you are all interested in. So with that, thank you and have a great evening.